Hey everybody. So we're moving forward. Hopefully going to finish today with the end of these two case studies, which were again talking about sort of physico-chemical properties and Lipinski and Weber and all those things. And then we did an introduction to some chemical informatics tools and computational tools to do drug discovery. Anyway, so yeah, we're going to do two case studies, one on the modeling and development of GP41 inhibitors, which are HIV fusion inhibitors. And we've talked about HIV fusion a couple of times in class as a drug target. And then we're going to move into it's a class of chloride channels called SLC26A3, and we have some videos that show molecular dynamics of these inhibitors. This is a drug target for constipation and I think like kidney stones also. All right. So what did we do last time? Just the last little step. I'll go back to the previous slide. We just showed the idea of in silico synthesis, which is sort of carrying out chemical reactions within a computer using free online tools. And we used this thing at the at UC Irvine. The program is called RDKit, and this is a openly available set of tools that can do a whole bunch of things. And they're conveniently available on this UC Irvine website, and it's at cdbics.uci.edu. And what we did in that last little bit of the last lecture was we, I kind of created little mini versions of these two functional groups, 1100 amines, 96 carboxylic acids, made them available on my website. And we used these kind of as substrates to build new molecules, new analogs of these Notch inhibitors. And Notch is one of the drug targets we've discussed is one of your homework assignments. And then we synthesized 96 versions of the amine component with different carboxylic acids. And we synthesized 1100 derivatives that use the carboxylic acid component, and then used a library of different amines. So yeah, we made a whole bunch of like analogs of these molecules using these tools. Okay, and so what we're going to do is maybe show one of the things you could do after virtual synthesis is to do like docking or some sort of computational modeling as a drug discovery step. We're not doing it for this project, but I'll show you a real project in a second. All right. Oh yeah, we can also, like I said, take the Lipinski and Weber properties of the molecules we make, because that's something you can do. It's a little harder to actually evaluate activity against a drug target, but anyway, yeah, we can't show everything. But yeah, okay, let's move forward. Okay, so we're going into case study number 1, modeling and development of GP41 inhibitors for HIV fusion. Okay, so let's get into example number 1, which is one of my earlier research projects. It's something we did maybe about 5 years ago or something. So we were identifying inhibitors of HIV-1 fusion protein GP41, and we'll review kind of what it does and how it's involved in HIV fusion in a second. Computationally, we do a couple things here in silico synthesis to make molecules that we could potentially evaluate, and then also using massive compound libraries, so on the order of millions of compounds as part of our synthesis strategy. And something we were able to do for free, essentially, obtain these massive compound libraries to build large virtual libraries of molecules. Okay, and then we did computational docking, which we talked about. So docking the virtual library into into our model of GP41, HIV fusion protein. I think this was also maybe developed as a homology model initially. And then we did a little bit of biological evaluation of some of the molecules we synthesized. I should say that we do a synthesis step here. So we actually, in addition to doing in silico synthesis, we did real synthesis of a subset of the materials. Okay. So, as we talked about before, HIV has several well-established drug targets. Here's another of those cartoons that are prevalent. And the idea is we'll start kind of from number 1 and then move all the way through this process. Initially, we kind of assume we have a mature viral particle, that HIV viral particle, that kind of it does a process where the virus interacts with the host cell and kind of associates with it in a way that the virus then enters the cell, and its viral RNA gets converted to viral DNA through reverse transcription, and that gets integrated into the host genome. And then there's basically then the host will synthesize more of the RNA and actually create like a new viral particle, the new genetics of a viral particle. And there are some proteins that get synthesized, and then the RNA gets resynthesized. And then it all kind of self-associates, which is amazing this all happens, and then creates a new viral particle. And that gets excreted, and then it's not it's an immature viral particle, and then through some processes like proteases, HIV proteases, the viral proteins get converted into mature proteins. Anyway, this is the rough cycle. We'll zoom into this fusion process in a little bit and talk about some of the specific steps that occur. Okay. And in terms of other drugs, we've talked about AZT or I think it's Vudine. That might be a misspelling. And this interrupts reverse transcription. So it's a nucleotide analog, and it basically gets incorporated into the growing DNA chain, viral DNA chain. And then it just truncates it and it kind of messes it up. There's some toxicity because this thing can get incorporated into your DNA as well. Then there's the NNRTIs, the non-nucleotide reverse transcription inhibitors, like nevirapine. And we talked about some of these earlier in the course, I think maybe in a homework assignment. And then there are protease inhibitors, so darunavir, which we've talked about maybe a couple times. There's indinavir, a couple other ones. And they kind of inhibit this protease cleavage process, the synthesis of mature viral proteins. And then there are fusion inhibitors, and these HIV fusion inhibitors kind of interfere with this process and with the virus fusing to the CD4 receptor, and then there's these co-receptors, CXCR4 and CCR5, that are kind of involved in this process where the virus gets incorporated into the cell. And this protein kind of just interrupts a protein-protein interaction between the virus and the host cell. And would you think that this would be a nice orally administered drug? And you know, because we just talked about Lipinski and Weber rules for oral bioavailable drugs, and this. Well, I actually looked up a SMILES string for this, and I couldn't find one because it's a lot of atoms, and SMILES doesn't work really well for these big peptides. But anyway, yeah, this is not orally bioavailable, and it's It's taken by injection. It even says it right there, for injection. And also even the container. I mean, this is an ampule that you would insert a syringe into to suck up the peptide solution and apply intravenously. Yeah, some of these proteins have nice X-ray crystal structures. This is HIV protease, which is one of the, in terms of structural biology, and we'll say structure-guided drug design, which means designing a drug that fits into the pocket, or you could say like a key that fits into a lock. HIV protease is one of the early success stories of this, because it was a crazy disease. We were just getting good at X-ray crystallography and having inhibitors bound to a protein and getting crystal structures of it. And then so we had an urgent need, and then this great technique, and we're really good at making organic molecules quickly. So yeah, some of the HIV protease inhibitors, it's just a really cool story, and how they've also improved. This is not a first generation inhibitor. There's multiple generations of this. So the earlier generations are definitely not as good. Okay. All right, so this is a sort of zoomed in representation of how HIV fusion occurs, and I think I have one more slide that even goes into more detail. But for now, the idea is that here's the viral membrane, the HIV viral membrane, and there's some proteins on the surface of it, including GP41 and GP120, and they have some other little loop regions in the proteins. On the bottom is a host cell. So what happens is that GP41 is a bundle of a couple different regions of peptides, HR2 and HR1, that kind of start getting unraveled, and the GP120 gets bound to CD4, which is a co-receptor on the host protein, on the host cell. Okay, and as this happens, other proteins get recruited, like CCR5, which is a co-receptor that interacts also with this sort of complex, and then through this process, GP41 gets basically inserts into the cell membrane, and then through some kind of mechanical opening process, the peptides of GP41 essentially tear open the membranes and create a pore, a fusion pore, which allows things from the viral side, the genetic material and protein machinery and things like that, to get inserted into the host cell. It's pretty amazing that this actually happens. Okay, and so GP41 is a key player in this whole process, and it actually does the kind of injection into the host cell. Is initially buried in the viral envelope, which is kind of in this process. It eventually penetrates the host membrane. It is exposed, and it unravels after GP120 binds to CD4 into the host immune. Well, and CD4 is a host immune cell glycoprotein. So it's a lot of glycoproteins are involved in kind of cellular recognition, and this protein's actually, it's part of the recognition process that the virus uses to sneak inside. Our strategy is to kind of bind small molecules, or we could do a peptide as well, but we're looking for like orally active drug molecules that will interact with GP41 and kind of maybe prevent the unraveling process, and also prevent the insertion process into the cell. Kind of like the fusion protein drug Fusion that we just saw. Okay, here's another depiction of how it happens. Okay, so now the one difference is that we we flipped it over. So the virus is on the bottom and the host cell's on the top. All right, so we got GP41 kind of buried in the bottom. We have GP120 on the top. And CD4 is this glycoprotein on the surface of the host immune cell. And there's an initial interaction, and then there's kind of a recruitment of this chemokine receptor, CCR5, and a couple other things like that. And this starts forming a tight association. And what happens then is these HR1 and HR2 components of GP41, so they're peptide components, they form this 6 helical bundle. So they self-associate in this kind of manner that physically like springs together the viral cell, the bottom part, and the host cell. So this creates a tight association, right? And then that actually tears a hole in the membranes and allows the viral material to go inside, and that's into the host cell, which is crazy. I mean, I'm fascinated by how this works. And you can imagine, like, where would we maybe want to install an inhibitor? And the answer is like right inside here, like in this little hole here, where we could insert a molecule that might prevent this tight association, right? That's the basis of HIV fusion and how we might use that to treat HIV. And, oh yeah, this is a great paper. It's called 30 Years on HIV Receptor Gymnastics, because this looks like a gymnastics move here, and the prevention of infection. So this you all can get. It's an open access journal. I think I put it on the iLearn. If not, I will try to remember. You can remind me also, or just download it. If you Google search this, you can get the paper. And this just again talks about kind of this discovery of HIV fusion as a drug target. Okay, so there's a couple of these steps again that involve GP120 and GP41. GP120 binds to the CD4 receptor, which we're showing. GP120 changes shape, increasing its affinity to the co-receptor CCR5. So some weird things happen to the proteins that cause an increase in affinity to the co-receptor. And CCR5 is this kind of magenta-ish thing, the chemokine co-receptor. And then GP41 unravels to reveal helices HR1 and HR2, which are the green and the blue thing. HR1 promotes membrane penetration into the host cell. It's little yellow arrows. And then this self-assembles to create a fusion pore, which transmits the viral contents into the host cell. And so this kind of hinge sort of collapses on itself, and now you have this 6 helical bundle, and this causes the two cells to get really close, and ultimately opens it up and allows the viral material to transfer inside. Fusion is a peptide that we showed, and it can kind of act as a peptidomimetic that will insert into this into the bundle and kind of prevent this nice 6 helical bundle from associating like this, and prevent HIV viral fusion. And that's what this peptide is. Okay, so this has a lot of resemblance. This peptide has a lot of resemblance to, I forgot if it's HR1 or HR2, but it basically binds into this complex and prevents this helical bundle from taking form. And our drugs, what we're trying to do is discover orally available drugs that would have the same sort of effect. And all that has to do is mess up the interaction just a little bit, and then this would stop it from working. That's certainly the theory. Okay, so in terms of our project, well, there's one more thing I have to tell you about that was done before our project, and this was by a collaborator at Scripps Research Institute, which is a major research university in San Diego and I think Florida also. And essentially, what they did, and they were doing this for a different project, but they made a big library of molecules for a different project, and then through collaboration they worked on they basically applied this big library of compounds against a different drug target. I forgot what it was, the second. But also then they applied it against HIV fusion, as just to see, like, could this possibly work as an HIV fusion method? Because these are peptidomimetics. What does that mean? It means it kind of looks like a peptide in some ways, right? So you have an amino acid here with a R1 group on it, peptidomimetic, right? So it's like it mimics a peptide. So, and then I say X and Y, but I really mean R1 and R2 here. There's 20 amino acid side chains on R1, or what we're calling X. And then you have a benzene and a nitro group, and then you have kind of like another side chain, and we're calling it Y or R2, and that's 20. So 20 times 20 is 400. So this lab at Scripps, Dale Boger's lab, who's a very, very major organic chemist, very accomplished organic chemist, medicinal chemist, and has done a ton of work in a lot of projects. Well, what they did is they made this 20 by 20 library, and then through another collaborator that I've been working with over several years, they screened this against GP41, HIV fusion. And what they found is that the best R1 and R2 substituents are kind of aromatic or heteroaromatic. So maybe like sort of phenylalanine type derivatives or tryptophan, things like that. And also for R2, like benzenes and maybe a naphthalene, like two benzenes attached to each other. Okay, these are easy to make, which is really cool. You can kind of start from this sort of derivative, fluoronitrobenzoic acid, and the first step is a nucleophilic aromatic substitution, which is so you take off the proton from the alcohol, so make it O minus. O minus attacks the carbon, and we saw how then the electrons either push onto the carboxylic acid or onto the nitro. And then they kick off the fluorine. It's a two-step process, right? Well, for 3 steps, I guess. Base takes proton, O minus attacks, and then the electrons get delocalized. And then the delocalized electrons swing back, kick off the fluorine, and then that just attaches these two, right? And then the other thing is that then we would attach an amino acid to the carboxylic acid and use maybe EDCI or HBTU, one of those things that you know about. And then if there's any deprotection, we may do that. Overall, though, it's pretty easy to make this library. And then the other thing is, I believe that when Scripps made this, they did this automated. So they were able to do this using a robot. So they didn't actually, I don't think they did all of these by hand, because that's, 400 compounds is a little bit of work, right? And I certainly, if I told my research students to make 400 compounds, they'd probably find another research lab. So any way you can use like robotics to speed up synthesis, that would be helpful. And certainly Scripps has the robots to do this kind of stuff. Okay, so this is the library that Scripps synthesized. So a lot of these are sort of non-natural amino acids, and we have the R1 group. So these are like real amino acid. Well, they're amino acids. Some of them are non-natural. Some are natural. Some are non-natural. So like they have like what's this? Leucine, threonine, isoleucine, valine, lysine. Serine, but then they have things like 4-chlorophenylalanine, They have this weird naphthyl amino acid. This thing's like a phenylalanine, but it's got an extra carbon. I think it might be called homophenylalanine. A couple other sort of weird things that they just added into the mix. So these are, but generally, yeah, these are amino acids with a couple non-natural amino acids. Okay? And then we have the other side, which is sort of the R2 side. And these are things that kind of look like maybe some amino acid side chains. But it's not an amino acid, right? This is off of benzene. It has amino acid character, and the side chains kind of, they act like an amino acid, but they're incorporated into a sort of a synthetic, non-natural thing, and that's why this is called a peptidomimetic. Anyway, so we have ethoxy, we have this 4-methylbenzene ethoxy, isopropoxy, etc., methoxy, all sorts of weird looking things, and then they have some maybe more aromatic ones, and naphthalene, which is the 2 benzenes and the CH2 oxygen. This kind of looks like a tryptophan, sort of like a tryptophan that would be attached at the R2 position. Maybe like a lysine type derivative with 4 carbons, 4-chloromethyl, etc. Okay, so yes, anyway, they designed this for another project, and then they're like, well, let's just try it out for this GP41 project, just for fun. And let's, seems like a reasonable thing to try. Especially once you make a big library, it's like, try to milk it for other projects. This is, I think, one of the papers. There's maybe a follow-up paper as well on this. Oh yeah, what's BMCL? It's bioorganic medicinal chemistry letters. All right, so this just shows a couple of the best inhibitors that they discovered. The ranking was a docking ranking, so let's ignore that. But overall, these are roughly sub-micromolar against GP41, which is, it's not nanomolar. Well, it is actually high. I would say high nanomolar, so kind of like the 800, 500 nanomolar range. And yeah, and so these are real molecules that they discovered that have GP41 fusion inhibition activity. Okay, so what are some of the good ones? Like naphthalene up top, maybe chlorophenyl on the amino acid, a non-natural amino acid. Naphthalene on the amino acid, which is totally non-natural. This is not a naturally occurring amino acid, right? Indol, kind of like a tryptophan, and all these kind of permutations. All right. And one of the things I did, and when I started this project, because none of this was done by me, I should say. Synthesis of these and also the biological evaluation was not done. What I was doing is I was looking at the computational modeling of this to maybe create a new project, which is what I did. Okay, so this just shows the kind of a general binding mode of these, and this is a homology model, refined homology model of GP41. And one of the key things in this structure, and you can't see it too well, there's a little blue smudge here. That's a lysine that I forgot what lysine coded is, but and there's a nitrogen of a lysine. It has some white H's around it. So my point is that this is a positively charged region of the molecule, and this seems to be a pretty critical amino acid binding point for all these molecules. So you have the carboxylate, which is negatively charged, and then this is a positively charged thing. And then it fits nicely. I mean, you have this sort of aromatic ring, sitting right there. The nitrobenzene portion is right here. Then you have the other portion of the molecule kind of digging into this pocket. And this looks good. I mean, right? I would think you'd agree that this is a reasonable, possible or sitting point for our molecules. Okay, so what did we do then in terms of a computational experimental approach? So this is kind of where my lab got involved in this. So we start with a previously established binding mode of these 400 inhibitors, and a couple of them are really good. Not all of them really good. And this is work that well, I say it's not done in my lab, but I did the computational part. I didn't do the synthesis or the biological evaluation. So yeah, kind of starting with this and these kind of compounds, we're thinking, well, let's do in silico synthesis. Let's create a virtual library of analogs using commercially available synthetic building blocks to virtually synthesize about 6,500 inhibitor candidates, right? And if you were looking at this molecule, because when you look at the wider library, what was, did it all look like this? No, you had all sorts of things, little alkyl and serines and ethyls and methyls and propyls, but very few of these kind of aromatic type compounds. So given this, we have a new hypothesis of how did these molecules maybe sit in here? Well, it looks like you need maybe an aromatic thing here, maybe an aromatic one there, right? And so what I wanted to do is maybe expand on this, right? Expand on different aromatic chains there, greasy things that have this kind of effect, greasy things maybe that have this effect. And then given the binding mode, which is sort of how these things sit in, we could screen them really quickly. So if I make 6,500 compounds virtually, well, that cost me $0. Then we can dock them, and that costs about $0. And then we can maybe find something cooler than this, different, and then maybe make some of them, maybe like 20 of them or something. That was my project, right? So the first thing we did is I was like, well, let's keep 4-chlorophenyl stable down here, right? And let's keep on the other side. Now we'll keep like indol. Like this is one of the good R2 substituents. We'll keep this solid, and then vary this. And basically, both of these things can be synthesized really easily because in the lab, it's really easy to make these molecules. But I could design virtual libraries. So a 6,000 compound virtual library with different alcohols attached, right? And a 500 compound virtual library that basically use different amino acids. Okay, as you can see, are we restricted by the number of amino acids? Well, there's tons of non-natural amino acids, and I can download them on my computer. So this is basically 2 libraries comprising about 6,500 compounds total. And then the idea is let's dock them. Well, we have a docking model, so feeding in these virtually synthesized molecules into the docking model is pretty easy. Replicate the same binding mode, determine some good scoring inhibitor candidates, and then just make some. Because ultimately I'm a synthetic lab. Why don't we just make a couple and then actually check them for activity? That was a little mini project. This is one of my master's student projects. We synthesized about 20 compounds total of the best from this study. And then we screened them against, we submitted them to our collaborator that actually does the biological assay, because we did not do this GP41 biological assay in our lab. A very brief summary of what it is they have a biochemical model of the protein, and I think it actually is the protein. I think they use GP41. And then they have a peptide that is attached to it or associates with it. It's a fluorescent peptide. And what these compounds do is they compete off the fluorescent peptide. It's something like that. That's from my memory, and I haven't looked it up, but it's in some of the papers that I just mentioned, the BMCL paper. Anyway, yeah, this is a cool little project we just kind of came up with. In silico synthesis. So this is showing in silico synthesis with something called Scitegic Pipeline Pilot. This is a really cool computer program. Nowadays it costs a lot of money, so just want to show it because it's kind of a cool visual tool. And there's other programs like this that are totally free right now. When I did this project, this is all there really was, and I think it was free for academic use back then, but then they got rid of that project, or that program. Okay, so we're doing in silico synthesis using Scitegic Pipeline Pilot. So what we're basically doing is, I think we were envisioning just taking like maybe an acid chloride of this component with the indol type thing attached, and then just getting a bunch of amino acids and doing this all by computer, where this gets attached, okay? And this makes the amide linkage, okay? And so I started with 6,470 compounds from a chemical company called Boc-chem. Boc-chem is known as a really good supplier of amino acids. So if we ever need tons of amino acids, this is the company, one of the maybe better companies for that. There are other companies that are kind of exclusive in amino acids. But you're able to get all this, the smiles. We're able to download all the smiles of all these amino acids right here. And then after I did this, we got about 500 compounds out that were suitable for docking. And yeah, some of them didn't work, or they were kind of weird. And yeah, it's a little surprising that we didn't get more compounds out. But 500 is, it's even more than the Boger lab synthesized. But that's what we got. And then we docked them, and we successfully docked about 95 compounds. And that was kind of exciting. So we got 95 compounds that kind of looked reasonable, like they interacted reasonably with GP41. This is this just shows what Scitegic Pipeline Pilot looks like. It's again, it's a cool idea. This program you can't use anymore, but there is a program called KNIME, KNIME, that kind of does the same thing. But you can sort of see what is going on here. Okay, so we'll step through a little bit of this. Here it's called a reaction reader. So this like reads in the amide forming reaction that we're drawing. So it's like a little file that's like, what was it called? The reaction file format, because we have smiles for chemical structures, right? What was it called for a reaction? Remember? It was called smirks. So this reads in a smirks file, okay? The amide forming reaction file, smirks. And then it basically reads in the smiles of a file of my 6,470 compounds from Boc-chem. So this read in the smiles. It did a little bit of filtering, a high throughput screening filter, which kind of removes maybe some nasty compounds. So you can see what happened. 5,800 survived the filter. 648 got excluded, and I just dump them out. So it's like it's a little pipeline, this little machine, right? It's really fun. I wished I could still use this and maybe teach with it. And all of these are little tools that process the data. Substructure count. I think that was looking for is counting the amine or something. Yeah, I did a couple steps that I can't remember. And then I read the smiles in of the other part. So I read the smiles in of the library and the smiles in of my other piece, and I just made that smiles up. It read in one smiles. And then this enumerated the combinatorial reaction. This performed the reaction. This actually formed the amide. And then these little tools kind of allows me to view the results. Whereas these little branches would take the products that I made, add hydrogens, because somehow the hydrogens were missing, and it converts them into three-dimensional coordinates automatically. This is all done, this little pipeline. Generated three-dimensional coordinates, and then it ionized them at physiological pH, so pH 7.4. So that would, it might deprotonate the carboxylic acid, for example, at pH 7.4. Then it minimized them in three-dimensional space, and then it wrote out a mol2 file. And a mol2 file is just another chemical drawing format. And this I fed into the docking program, right? So I was able to do all of this synthesis, do some preparation, and then I basically dock. I docked with the mol2 file. Okay, so that's pretty cool. Then this just shows the other variation of the molecule. So now it's like we'd start with the maybe an amino acid attached here, and the fluorobenzene part, and then our virtual synthesis now is going to be reacting this with primary alcohols, and then the primary alcohols would go through that SNAR reaction. Remember, nucleophilic aromatic substitution. So the oxygen would kick off the fluorine and make the ether linkage. Okay, and here I started with the enamine and Cambridge commercial libraries, which is 2.3 million compounds. When I did all of this, I got out about 5,000 compounds, because not all of these million compounds were alcohols, for example. So, but I started with this library, and then when I pushed it through, it reduced this size of this virtual library to about 5,600 compounds. And then we docked them, and now we got about 585 successfully docked compounds. So this was a whole bunch of different compounds, where the variation is on the oxygen CH2 in this R2 group. Okay, we did the same exact kind of thing. The only difference was the SMERX file, which is the reaction file. It was different because now it's like it's a nucleophilic aromatic substitution, so the SMERX file is different, but for the most part this is the same. All these numbers are wrong because this is for the other one. Just wanted to show that this is a really cool approach to do virtual synthesis, and also do other things like three-dimensional compound generation and ionization. They have these filters, the HTS filters. Some of them do like Lipinski filtering, so I can take my final molecules and do Lipinski filtering of them, and to make sure they pass the Lipinski rules. That's the kind of stuff this does, and it does it in a really cool visual way that is fun to do. And then you get, you feed your stuff in, you generate your output. Okay, and again, the other program that's kind of like this is called KNIME, KNIME. There's some YouTube videos on that. If you search for KNIME drug discovery or KNIME chemistry, because it's used for other things as well, you might be able to find some more information about that. Or we could talk about KNIME. If you're interested in KNIME, I can probably we could actually step through it and build a little machine like this. Okay, but yeah, it's like a little machine builder. Computer machine builder. All right, so this just shows some of the ones that I discovered through after I did my virtual synthesis of a big library, looking for kind of weird non-natural amino acids, for example. I found like this one, which had the tryptophan buried in there, and there's the carboxylic acid talking to the key lysine. This is a weird pyrazine. Totally non-natural amino acid type derivative. Here's a quinoline, which has a nitrogen on there. It's kind of doing the same thing. S-benzylcysteine, so it's a cysteine, which is CH2 sulfur with a S-benzyl group on it, and so it has phenyl, CH2 phenyl. Yep. Boc-protected L-ornithine. So this is another kind of example. Ornithine is like lysine, right? So it's CH2, CH2, CH2. But lysine has 4 CH2s. Ornithine has 3. So it's CH2, 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 and it has a Boc protection. So NH carbonyl, and there's a Boc group. So this one happened to also dock really nicely. Very quickly, just to wrap up this project. We're showing Ki against GP41 binding competition assay. So Ki is roughly the same idea as IC50. It's just another measurement of activity, and lower is better. So nice small numbers are what we want. And my collaborator did this work, so I wasn't involved in this. But this shows ultimately what we synthesized. We kind of designed a synthetic project that looked at different well, I'm not showing I guess what I'm not showing is the central benzene thing. But this is showing the site. Oh yeah, I am. I'm doing that. Yeah, these are all amino acids. Some of them are weird ones, like this iodotyrosine and dibromo-tyrosine, dichlorophenylalanine, cyclohexane, methyl cyclohexane, which is kind of like phenylalanine, right? Phenylalanine, but with a cyclohexane, non-natural amino acid. This is a benzothiazine that's kind of like indole, but it's a sulfur. This is a biphenyl, so it's got a big benzene out here. Very weird, yeah. This is methyl cyclopentane, and then we have S-benzylcysteine down here. Up here, we did in this case, we just did 3 variations: the indole one, the naphthalene, and then the chlorophenyl. Okay. Of these, the best one appeared to be this one with the lowest Ki value, 0.2 micromolar, or about 20 nanomolar. So it's actually a pretty reasonably potent one, with the naphthalene on the other side chain of the virtual side chain of the inhibitor. The other 2 did not work out as well, and looking at other ones, in general, the data is kind of all over the place. There's not a lot of rhyme or reason to this, but I guess in terms of general activity, I don't know, maybe this dibromotyrosine one, we saw pretty good activity with the 3 different variations off the benzene. Still kind of micromolar-ish, maybe a little more than micromolar. This was our best compound. Okay. Anyway, yeah, that was a kind of cool little project. This is not we didn't turn this into a clinically available drug. We didn't really do much more with this project. This is just a master's student project, but we did get some biological data. So I'd say the approach kind of worked, and we did discover a potential new GP4E1 inhibitor. All right. So this is student work with my student Michael Pun. He got his PhD at Washington State University in Pullman, and then Miriam Gotchen, who's a professor at Touro University in Mare Island, kind of Vallejo-ish area. So we're still planning to publish this. We haven't had a chance to yet. Okay. Okay. So then I have a little bit to talk about another project I'm working on, and currently with one of my master's students who's graduating probably in the next month or so. Inhibition of a protein called SLC26A3, which is a chloride channel. It's a chloride antiporter, so it absorbs chlorine in one direction and excretes bicarbonate in the other direction. Has some drug relevance as a drug target for a couple of diseases. Okay, so what about it? So this is a huge project. I'm only going to focus on the computational aspects of this. We discovered a inhibitor scaffold called 8-chlorocoumarin that inhibit this thing called SLC26A3. It's also called DRA. It's a drug target for chloride absorptive constipation and kidney stones. And there's a lot of biology about how this works. My student has basically been synthesizing a whole bunch of analogs of this 8-chlorocoumarin. I'll show the structure in a second. Computational wise and relevance to this lecture, there's a little bit of homology modeling, docking, and molecular dynamics. And we also have a lot of biological data, but I'm not going to really go through that in detail. I'll show the best result. Also tends to synthesis. Most of this is a synthesis project. So yeah, we did a ton of experimental work, synthesis of a library of 20 compounds, biological characterization. The actual synthesis is probably around 50 compounds because it's a multi-step synthesis. Each of the compounds had to be characterized by NMR and mass spectrometry and all sorts of stuff like that. But anyway, and yeah, so what we're showing, this is a summary of a ton of work. This is showing a dose response curve in IC50 values for a kind of like what our starting point was. It's called the previous lead molecule, SLD26. IC50 was about 40 nanomolar. And so when we say log concentration, what does that mean? Well, -1 is 1 micromolar. -2 is 1 nanomolar, right? So this is about 1 micromolar and this is 1 nanomolar. Remember that percent inhibition is on the y-axis. And so IC50 is referring to the halfway point, the 50% point. So it's kind of like there. And in concentration terms, when we do this, we usually don't think about logarithm of concentration. We just think about an absolute value. And anyway, so at this is the new molecule we just that we discovered, along with other molecules I'm not showing. This is the best new molecule, but its IC50 is 17 nanomolar, right? So if you go to the 50% point, you go down and you transform the logarithm. It's 17 nanomolar, which is happens to be the most potent compound on the planet that we know of this protein. And this is our starting point. So the main thing that we did in this project was we did a whole bunch of variations. We synthesized a whole bunch of variations of this molecule. A lot of variations, like putting a chlorine into this central part of the molecule. We did bromine. We did fluorine. We did variations of stuff out here. We moved the methyl to the other side. All sorts of stuff, and again, we made a bunch of molecules, screened them, and then this was like the best compound. Okay, without going into detail, which I probably could have done, but I'm trying to focus on the computational work. Okay, so this whole project was really non-computational, but recently we did a little bit of computer modeling, and that's what I'm trying to show you. Okay, there's Maria, my grad student, who did all of the synthesis work and a lot of the biological screening aspects of this. Okay, so computational approach to determine a putative mode of binding. So that's kind of what one of the recent things we've done actually since the COVID-19 outbreak. We're trying to kind of work remotely and do some computer research while being quarantined at my house. Anyway, so this was SLC26A3. This was the human protein amino acid sequence, and we actually did this in class, maybe, or in the last video lecture, where we built a homology model starting from the amino acid sequence. But I used a program called Yasara, which takes a lot longer than the one we used in class. We used Swiss Tools, which was a Swiss Tools was the free one. It's a free web-based one, but what I did for my research project is I used Yasara, which is maybe, I want to say it's a little better maybe, and a little more exhaustive, and it does some more refinement of the protein structure. Anyway, so that created a tentative protein structure. So it looks kind of like a chloride channel might look with this collection of alpha helices. I embedded this into a simulated membrane lipid, into a membrane lipid using phosphatidylcholine, which is a bilipid membrane component. Okay, it is a chloride channel, and from this angle it looks like it doesn't go out the side of the membrane, but it's actually kind of the angle that I drew this at. It does, the protein does actually make it all the way through the lipid. Okay, so what I then did is I took this and I did computational docking with all the molecules we made. And I think we made about 20 molecules in our synthetic library into a binding pocket in the cytoplasmic surface. And so there's kind of like two surfaces of a channel protein like this. There's extracellular, which is the outside of the cell, and the cytoplasm, which is kind of the inside of the cell. So the bottom is representing the inside of the cell. And we actually found a docking pocket, like a place where molecules could fit in. And that's where we docked. We docked our inhibitor. And so this kind of shows, well, from this vantage point, kind of where that where that molecule resides. And this is our new potent lead compound. So it's this kind of yellow thing, and it found a little docking pocket, and we were able to dock all of the compounds in there. They all fit. It's like a little hole, and all of the molecules that I docked were able to fit. One of the questions that was not totally clear from docking was whether, like, you have two ends of this. Does the carboxylic acid kind of poke upward, or does the carboxylic acid kind of poke downward? You'd think the program would just find that perfectly well, but docking doesn't really look at dynamics of proteins, and it just looks at a static representation of the protein. But anyway, yeah, so there could be two orientations: carboxylic acid kind of going upward, or carboxylic acid kind of going downward. And either way, it fits. So how do we figure out which orientation might be the correct orientation? And this is where we're extending computational docking to molecular dynamics, to study the orientation of binding and the stability of the protein ligand complex, and also the energetics of the binding. So molecular dynamics is kind of, we're using it as a follow-up tool to use after docking, to kind of look at, like, watch the protein equilibrate in this crazy membrane environment. And there's also water. There's water on the outside. There's water on the inside. Water on the outside. This is a very complicated system, right? And we can envision and visualize the energetics and the binding energy of our molecule. And this will help us figure out our question: which side goes in? Carboxylic acid going upward, or does the trifluoromethyl go upward? Okay. All right. So, without going into much more detail, I just want to show a couple movies, because this is cool. These are fun little movies. All right, so I'm going to navigate here. I have a couple YouTube videos. You can watch them also. Remember, if you want to copy and paste these, you can get these off my PDF files that are on the iLearn site. But I'm going to play the movie. It's on YouTube. And the first movie I'm going to show is a zoom out. So it's not that it's kind of looking really far away, and what it's doing is it's showing how dynamic this system is, with everything equilibrating. This is a massively complicated system. You have every one of these lipid molecules are equilibrating. You have the protein, you have the water, you have the drug, all of this stuff. So what I'm trying to do is show you how crazy this really looks, and what molecular dynamics is really doing. So, okay, so here's the movie, and this is showing the proteins equilibrating in the middle, the drug's the yellow thing, all of the water is going all over the place. The lipid, I'm purposefully making invisible, so you can't really see it too well, but the lipid's right in the middle of this thing, and all of those atoms are equilibrating also. And so we're doing this over a, whoa, haha, sorry. We're doing this over a large period of time. And like a normal molecular dynamics simulation is measured, like what's the lifetime of like a drug binding to a protein. It's in the nanoseconds, kind of the nanosecond time scale. I'm simulating about 50 nanoseconds or so, which is actually a lot of time. And what I'm trying to do is figure out, which orientation of the drug is kind of favorable, right? And so 50 nanoseconds is reasonable for that. How long did this simulation take on my computer? Well, I have a couple of special computers that I use for this, and it took about 7 days of continuous computation. to do this. Okay, so what's the next one? The next one is a potential inhibitor, a potent inhibitor bound in a postulated bad orientation. So I'm going to show a kind of a bad orientation, and I'm going to show a good orientation. So this is the second movie. Now we're going to actually zoom in and see what the drug's doing to the protein. Okay, so let's get in there, get that YouTube link, and I'm going to paste it and get it going. Okay, so now we're actually seeing how the drug might interact with the protein. This is really cool. This shows the binding pocket that I was talking about. The coumarin part, let's go back a bit. The coumarin part is sort of the central part of this molecule. And there's the carboxylic acid. And what I was saying with this bad conformation is the carboxylic acid is not really doing anything. It's just sort of sitting outside, kind of poking away from maybe the binding pocket. The binding pocket's kind of a little deeper in there. And what we postulated is that the carboxylic acid, which we know is important in the activity of these things, is probably doing something maybe more substantial and maybe interacting with an amino acid. So that's why we kind of think, oh, the carboxylic acid is probably deeper into the pocket. Okay, so let's see the next video. The next video will show the carboxylic acid kind of binding deeper into the pocket. Okay. Okay. All right, so this is kind of like what I'm leading to our hypothesis. There's actually an arginine, it's called arginine 164, that we think might be a crucial interaction with the carboxylate. So this is the carboxylic acid part as a negatively charged species, kind of poking outward. And this would not make a favorable interaction if it's kind of poking this way, right? The inhibitor carboxylate isn't positioned to make contacts with the polar amino acid residue, arginine 164. So then the good inhibitor conformation will be the opposite. And we actually have a pretty strong evidence now that this is bad and the other one is actually good, and we have some energetic, like a binding energy determination from our simulation that might show that. Okay, so let's go to movie number 2. Actually, it says movie number 2. I think we're movie number 3 now, but okay, let's go to this video now. I just said to movie 3. Okay, so there's the last one. All right, so this is pretty cool. Let's see. All right, what we're showing here is there's our arginine 164 I was talking about. And these little white lines are measurements of the distance between arginine 164, the hydrogens, the 2 hydrogens, and then the 2 oxygens of the coumarin carboxylate, which are these 2 oxygens. And notice the distance is really far. It's like 7 or 8 minutes. Or I'm sorry, angstroms. That's a pretty long distance. So that does not look that promising, right? So we just kind of let the movie go, and I was at first I was like, well, that's not really showing maybe the effect I want. I want this to be really close, right? So as the protein is massaging itself and the drug is kind of helping massage the protein, there's a dynamic interaction between the the ligand and the protein, right? And so what's crazy is a little bit later in the movie, which is about maybe coming up, the molecule, it almost like snaps and gets really close up. So it looks like there's some resistance. It's like it's encountering resistance, it's kind of smushed away, and then it just shoots up to the arginine, which I think is super cool. It's a really neat kind of dynamic view of how like a drug might interact with a protein pocket. Okay, here it's gonna come. It's still 8 angstroms away. It's still far away, right? So far, 8 angstroms, 8, 9, 8, 8, 9. So far, and then somehow it's gonna get really close. So smaller numbers. Look at that now. It just kind of shot up, and now we're at 1 or 2 angstroms. That's a very tight interaction. So it's like an encountered resistance, right? And we had a little energy hump to get over, and by letting the protein and the ligand equilibrate, it allowed this to happen, right? Because it's a dynamic system, and it's a very complicated dynamic system because there's all these amino acids and a lot of barriers. There's a lot of barriers to just letting my molecule slip up there. What are all these other little things? Well, these are other amino acids. I'm not showing all the parts of every amino acid, but these are little fragments, a couple atoms of the different amino acids. Okay. All right. So that was cool. And that's kind of what I was trying to show here now is as a little cartoon diagram of what happened is like initially the ligand is kind of stuck and far away, right? Maybe 8 angstroms away. But then with MD simulation and maybe around 5 nanoseconds in this little window, in this little time period, it induced a fit, which means that the protein adjusted its structure to allow and accommodate the ligand, and so then it shoots up and gets really close. Now we have a tight interaction. So yeah, anyway, that was a kind of cool little demonstration, I think, of what you can do with MD simulation. MD is a pretty tough technique to do. It's not an easy one for us to demonstrate, have you guys do, but in your research career, and if you move on to do other advanced chemistry or computational chemistry, this is definitely an area that you can kind of get into, and kind of learning how to do these molecular dynamics simulations. And it's kind of a computational heavy project, so you have to be pretty comfortable with computers. I think we are done for today's lecture, and I think we're actually done with this unit in the course that was talking about computational tools. The next thing we're going to do on Tuesday is get into prodrugs, which is going to be, we've learned about prodrugs. It's drugs that kind of get activated somehow through a metabolic enzyme and become, well, that's part of their activation process, is a metabolism step happens, and then it becomes an active drug in the body. That's actually a pretty substantial topic. There's a lot of examples of it, really cool examples. And so, yep, that's where we're going next. All right, have a good weekend.